So I will have a mix here, some background information and a little bit of our own contributions, because Tgf-Beta is simply a huge field and it is impossible and quite silly to pretend to present this into 30, 40 minutes. So I'll try to have a brief overview of the fundamental principles for, I hope, a broader audience. And wait, because I need to know what slides come next, because this is mostly not my science. Okay, fantastic. So, a brief overview of what Tgf-Beta signal transduction means. If you take a look at the pathway, what is remarkable, it is that it is very simple. You have several receptors, a bunch of ligands, but very few numbers of transducers of the pathway that are called as MEDs. Now, what is very obvious if you compare to other pathways that you know and that you will hear about today is the fact that you go from the cell membrane to the nucleus with a simple step, one unique step. The SMADs gets activated from the receptor, enters in the nucleus, and activates target gene transcription. And despite this remarkable simplicity, what is striking about this pathway is the huge variety of gene responses and biological effects that this pathway can trigger. So one of the points that I would like to address is how can we introduce complexity into this pathway? And so I'm going to give you a few examples, few ideas of how specificity can be dictated in terms of cell type dependency, such as different cell types turn on different gene expression programs. Signaling context. The same cell changes the responsiveness according to other inputs. As well as, very intriguingly and particularly investigated in terms of embryonic development, is the capacity of this pathway to trigger responses that are strictly dependent on quantitative amounts. Quantitative in sense of intensity of the pathway and of duration of the pathway. Of course, several mechanisms regulate each of these aspects. Now, in the evolution history, Tgf-Beta appeared with the multicellular organisms, suggesting that this pathway is in fact involved in the control of complex body pattern and homeostasis of complicated organs. Let's take now a better look to the core signaling engine. Now, mature ligand causes bridging of preformed dimers of type 2 with type 1 receptors to form heterotetrameric active receptor complexes, this stuff here. Now, the type 2 receptor kinase is actually constitutive active. And when it is bridged in proximity of receptor type 1, it triggers type 1 phosphorylation in this domain called the GS domain, which is a segment of the receptor just below the plasma membrane. This unmasks receptor activity of the type 1 receptor, which then triggers phosphorylation of the professional transducer of the pathway that are called the SMADs. Now, this is a very old picture, but I think simplified for today, but makes the point of what does it mean C-terminal activation by phosphorylation of the SMADs. Normally, the SMADs are in strange stoichiometries. Let's say they are heterotrimers or dimerized between each other. They are made of an MH1 domain, so-called, that mediate DNA binding, and a C-terminal MH2 domain that receives phosphorylation from the receptor, and also is very important for association with SMAD4. It's likely that in the unphosphorylated form, the SMADs are self-inhibited, whereas phosphorylation kinds of open them and renders them competent to activation. Also, a very important background information, although we generally tend to speak about the TGF-beta pathway, please remember that we should really distinguish two main branches of the pathway. One is the TGF-beta per se, that is transduced by type 2, type 1, and SMAD2, SMAD3 as professional transducers. And the other is the BMP, bone morphogenetic protein pathway, that is triggered by different receptors and different SMADs. There is only one common transducer, and this is SMAD4. Now, let me now start with taking you on the complexity of the TGF-beta pathway, starting from the first step, sometimes neglected, but this is ligand processing. Now, you should not think that TGF-beta comes in the same way you buy it from Sigma. There is a huge complexity in the secretion and production of these cytokines. In fact, a key event is the fact that the TGF-beta are produced as propeptide, inactive propeptide, that has to be clipped in this domain by a class of proteases called furins. And only this is generating the mature TGF-beta ligand, that is the active hormone. Now, what we discover is that we have clearly an important step already in the maturation, sequestration, and presentation of TGF-beta that, of course, set the rate-limiting step of the whole pathway. For example, there is a family of proteases called the amylin family that basically sits across the clipping site of the furins and prevents this maturation. So there is kind of a limiting step and regulation already very upstream here. In general, let's say that once the furin cleave the mature peptide, TGF-beta is certainly not active yet. It is latent. Actually, the majority of TGF-beta in our body is not in the active form, but is as a latent TGF-beta bound to the extracellular matrix. It is, the matrix is a sink for TGF-beta. So how can we take it out, ferreting out TGF-beta? Well, this is mediated by specific extracellular matrix proteins, such as thrombospondin, as well as by a variety of stimuli, including mechanical stimulation. Imagine heart, blood vessels, you know. These are very dependent on TGF-beta, and clearly there is a lot of mechanical inputs there. And you will have a talk here by Maria Celeste Aragona telling you about how cells perceive mechanical cues. And then acidification, oxygen, reactive oxygen species. And of course, this is very important for cancer, secretion of matrix metalloproteases that basically dissolve the matrix, taking out TGF-beta. And then TGF-beta, of course, works and depends on the tissue, the response. In the amylin 1, which is one member of the family, we discovered that this is essential for vascular smooth muscle cell growth, extracellular matrix homeostasis, vascular remodeling, altogether contributing to generation of vascular blood pressure. And then another macroscopic step of regulation, extracellular ligand trapping by secreted antagonists. And it is for me, historically, a great pleasure to start always this type of presentation with one of the best experiments ever carried out in biology, which is the Spemann organizer experiment. Now, we are in Germany. We are in the mid-late 20s, so 80 years ago. And Hans Spemann, a professor there, decided to understand the mechanisms. Of course, mechanism for us has a lot of meaning, but for him, you know, we had no protein, no DNA. For him, mechanism meant manipulation for what we could do. So he realized that tadpoles, of amphibian tadpoles, developed from complex morphogenetic movements that start from this little lip, which is called the dorsal lip. So he started to make transplant of pieces of the embryo into another one. And remarkably, what he found, that when he transplanted the dorsal blast upper lip, what he could be able to see was the generation of a second, full, perfectly harmonic body, a twin. Now, Spemann was, of course, a genius, and he had no lineage tracer, no marker, no ROSA 26 at that time, but he simply repeated the experiment, taking an albino and pigmented frogs. And what he realized that is the majority of the second body that he could induce in this way was actually not coming from the graft, but were changes in fate of the host. Of course, these new bodies, induced bodies, let's put it this way, entailed the whole secondary nervous system. And these attracted, generation of embryologists. And the understanding of the Spemann inducing factors have been for decades a holy grail for embryologists. And so cell biology in general, our own idea that cells talk to each other, that they induce each other, which is obvious to us, actually comes from this guy. And of course, this was a very well-rewarded Nobel Award to Hans Spemann in the 38, before the beginning of the war. So since then, there was a quest for understanding what are these factors. But this, everybody fails. It was only until molecular biology, and this is a molecular biology meeting, entered big time into embryology, that people were able to clone factors, secreted factors, emitted from the Spemann organizer. And with a major surprise, what came out was that these factors were not actually something that was signaling. These were killers of signals. And these specifically, the organizer, what came out is a source of Tgf-Beta extracellular antagonists. So basically, there is these molecules, such as chordin, that binds to BMP4 and it's not signaling, it's preventing signaling. So the system is amazing because we have a localized source of an antagonist that diffuse from one center toward the embryo, making, if you want, a gradient of antagonist that impinge on a relatively homogeneous distribution of the real signaling factor, generating a complementary inverse gradient, and cells are perfectly capable to feel differential amounts, I stressed the quantitative issue here, of BMP in this case, and generates all the different histotypes and diversity that you can see in a transverse section of any vertebrate body. Ventral island on the ventral side, then the splanchnoplura and somite, notochord, and so forth. Amazing. Now, this is not only that, not only patterning along this one, but the whole body is generated by gradients of Tgf-Beta, different Tgf-Beta, and their antagonists. Not only along these axes, but even in the antero-posterior axis. This is a cover of an old paper in Nature showing basically a tadpole with a second head coming out from the belly. And what is this? This is a gene called Cerberus that is basically a multivalent antagonist of different Tgf-Betas, and as well as Wnt. So I was telling you about this amazing Spemann experiment and the idea that the organizer is a source of secreted antagonists. And let me continue this with something I think very profound to close this embryological part of my presentation that Spemann wrote. And this is the term organizer rather than perhaps determiner. Is supposed to express the idea that the effect emanating from these preferential regions of the embryo possesses all the enigmatic peculiarities that are known to us only from living organisms. I think this is amazing that something this was written in the 24. Of course, the way I see this was that Spemann was meaning that the embryo that emerged out of his grafts is clearly able to generate a perfect body in its own proportion. So here are some thoughts on what I still think are unsolved mysteries concerning the extracellular regulation of TGF-beta ligands, because this is the part of the seminar I was telling you about. That is the following. What is actually blending, harmonizing the levels and range of activity of the distinct ligands and their antagonists? What is making them in the perfect proportion, such that all the body parts develop harmonically in perfect proportion with each other? How can the head and trunk organizer mutually coordinate their development? And what is making embryonic development so robust, meaning so able to withstand fluctuations, perturbations, insults? So it is actually, ironically for me, that one of the fields at the cutting edge of system biology is in fact something so complicated, such as building a body. And people, in fact, have been worked on these stories, developing, all sorts of regulators of the regulators, antagonists of the antagonists, all sorts of these complicated loops. But the general picture is emerging, and these are self-organizing systems. Networks able to constantly readjust each other, a set of interlocked feedback and feed-forward loops, opposing each other, but at the same time reciprocally adjusting each other's strength and activity range. So, closing up this part of the talk, I would say that the key lesson we learn from the embryo is that to quantitative inputs correspond profoundly qualitative answers, leading to the idea that TGF-beta are morphogens. Now, since I hope I could convince you with these examples that quantitative regulation is so essential, now we have remained only in the extracellular space. Now we have a big problem. Since extracellular space is fine, but what controls cell fate is in the nucleus, is how can we make a proportion between this tiny variation so profoundly important outside of the cells need to correspond to extremely dynamic interpretational capacities within the cells. So what key proportion between extracellular world and intracellular smart activity? And this is how basically transmission of signal strength and duration is realized. This is basically the concept of SMAD's nucleocytoplasmic shuttling that has been pioneered by Caroline Hill and many others. So basically the concept is the following, that SMADs are inactive and continuously enter and exit the nucleus. And these are in blue. Then active SMADs that are activated by the receptors, then enters into the nucleus, but their export is strongly impaired. Perhaps because the nuclear export signal is masked, or because there is an interaction with SMAD4. Now, these are basically accumulates in the nucleus, where constant inactivation, for green it turns back to blue, occurs. So there is a dynamic, a constant dynamic. You activate the SMADs and you inactivate it in the nucleus. Why? Well, this has profound logic. Because if you now inactivate the SMADs, this is forced to go back in the cytoplasm and ask again to the receptor, are you still on? And only if the answer is yes, and this is a quantitative answer, then SMADs can reactivate it and a new cycle starts. So this is a perfect way, keeping dynamic the nuclear complex, to maintain proportion between fluctuation, let's say a decrease or an increase of extracellular receptor activation with the intracellular amount of nuclear SMADs. So there is an important dynamic signal. So this keeps the cells responsive to changing signaling intensities. So once in the nucleus, the SMADs form trimeric complexes, one SMAD4 and two receptor phosphorylated SMADs. You see that there is this basic surface pocket that accept the phosphate in this configuration. There are thus three interfaces, A, B, and C. And this complex is the active transcriptional complex, is receptor phosphorylated and nuclearly accumulated. But as I told you, we need to constantly think to mechanisms that actually disrupt this complex. And it's the only way. We don't want to have this one accumulating linearly over time. No, this has to be kept proportional to the extracellular concentration of ligand. So basically we have to think to mechanisms that constantly inactivate, that generates instability into this complex formation. And of course, the first thing that comes to your mind is the possibility to revert what the receptor did, basically dephosphorylating the receptor SMADs. And in fact, phosphorylation, the phosphatase, the nuclear phosphatases are very important to turn off the pathway. But the point is that the phosphatases works only on monomeric SMADs. They are unable to work on the ternary active complex. That means that they clear up the receptor SMADs only after something else is disrupting the ternary complex. So we have been working on this and identified a SMAD complex disruptase, and this is mediated by a different type of post-translational modification, and this is, in fact, monoubiquitination. So we found that in the nucleus there is a ubiquitin ligase called ectodermin T1 gamma that is basically ubiquitinating key residues of SMAD4 here in orange. Now, if SMAD4 is ubiquitinated, it cannot make anymore an active complex, and therefore this disrupts the complex, setting free phosphoreceptor SMADs and ubiquitinated SMAD4. Now, in the cytoplasm, this cycle is reverted by the activity of another enzyme, which is the alter ego of ubiquitinases, which is called deubiquitinases, or DUBs. Just to give you the point of this, this is the real structure of the ternary complex. There is this critical interface between receptor SMADs and SMAD4, and the residues that are ubiquitinated, work by our own work and work from Aris Mustakas in Lund, found that this is mediated by ubiquitination of two fundamental residues, K515, K519, and basically you see the location of these residues. Once the receptor SMADs, here SMAD3, binds to SMAD4, they enter into a canyon, very close canyon, that makes this complex. Now, if ubiquitin occurs at this residue, of course, sticking a very complicated peptide like ubiquitin is completely incompatible with formation of the active SMAD complex. And this is basically disrupts complex formation. Now, I described this cycle of ubiquitination, deubiquitination as a mean to introduce dynamicity in the pathway. But this is not only cycle. We have just described, and this is unpublished, another cycle that is actually regulating the activity of the receptor SMADs by regulating their capacity to bind DNA. So there is actually another cycle of ubiquitination and deubiquitination that happens into the nucleus and basically hit one critical amino acid of the DNA binding domain of the receptor SMADs. And this again is ubiquitinated during transcription and then reversed by another DUB. So this creates competence at the level of the DNA binding other than what we described so far, complex formation. So there is an intriguing crosstalk between post-translational modifications of different sorts, hitting different SMADs in different domains, all together contributing to heterogeneity. Now, this story for us was very important because it allows us to tackle the dark side of the moon in TGF-beta field. I tell you what's the problem. The problem is that we have 30 ligands. TGF-beta is in the blood. TGF-beta is basically produced by many cells. It's accumulated in the matrix in vast amounts. And yet the responsiveness of TGF-beta is extremely cell-specific. How can that be? Receptors are ubiquitous. SMADs are ubiquitous. It's very obvious that we must have a macroscopic way to negatively, to negate SMAD activity, even when the signal comes. So this was the first view of a negative core element of the pathway that I think was important because it allows us to define what are the riverbanks, what are the components that actually take shaping TGF-beta responsiveness, such that, signals is canalized, allowed to signal only on a narrow confined spaces, but for the rest is confined by molecular riverbanks. And this is important because so far we have been knowing only what happened in knockout of receptor ligand and SMADs, drying up the pathway, but we had limited understanding of what happens when the riverbanks breaks, and actually cells that are normally not allowed to signal are now receiving signals. What is the biology of too much TGF-beta is actually enigmatic. So all this poetry is to introduce a couple of slides on the ecto knockout paper story. Basically, doesn't take much. You don't need to be an embryologist. Just let me tell you that these are extremely tiny embryos. These are a few days after fertilization. It is basically a sac in which the pluripotent cells are here, that are derivative of the famous inner cell mass cells, or ES cells. And in blue, what you see is activation of TGF-beta target genes. And you don't need to be an embryologist to appreciate that this is massively upregulated once you knock out these negative inhibitors. Massive. Such as this changes completely the embryo and the embryo dies. Which was great, of course, but didn't allow us to investigate what happened in the embryonic part a bit later. So we did epiblast-specific, stem cell-specific, embryonic stem cell-specific knockout of the gene. And what we found is, again, remarkable phenotypes. The bottom line of this finding was that in the absence of regulative monoubiquitination that I've been describing you, all the TGF-beta responses are upgraded to the maximal levels. There is no control. Everything goes to 100%. And so new phenotypes that were never observed before start to appear. So in blue, what you see is the homologous domain of the Spemann organizer, only that it is in a mammalian embryo. In blue, it's that domain of the embryo. As you can see, this is massively expanded. Basically, the whole embryo is now becoming a dorsal type of tissue. And this is, for instance, part of the a bit later organizer that in the ecto knockout becomes duplicated. You have two organizers. It's too much signaling coming out. So really dramatic type of phenotypes. So in the second part of the seminar, what I would like to talk to you is about TGF-beta signaling in adult tissues, where we see a different type of paradigm at work. What we found is basically we have one pathway, but multiple transcriptional programs. And this is a kind of a paradigm for many growth factors in which you have context dependency of activity. This is a pleiotropic role of TGF-beta in adult tissues. Clearly, TGF-beta is able to turn on different responses in different cells. It has, regulating migration and morphogenesis and growth of the endothelial cells. It is a very powerful immune suppressor. It is a pro-proliferation factor for fibroblasts was a potent growth suppressor for epithelial cells, and so forth. So how can we make sense? One pathway, so many different outcomes. And this is a simplified view of what is going on. Basically, the secret is that these meds are very poor transcription factors. They bind DNA in a very lousy way. In fact, what they need to turn on transcription is help from additional DNA binding cofactors in order to achieve high affinity and selectivity to specific target genes. And this modality, therefore, explains three cardinal features of TGF-beta. One is the pleiotropy. The second is the coordination. The third is context-dependent. Pleiotropy, of course, because once during evolution, one transcription factors evolve the capacity to bind to meds, then, the all target genes regulated by that transcription factors becomes TGF-beta regulated. And this, of course, has, a logic, and there is a coherent transcriptional program that explains the coordination. But as you well know, different cofactors are expressed in different cells, such that different gene expression program can be turned on in different contexts. And this depends, of course, on other signals and according to the cell's own history. Now let me tell you a few examples that are the most well-known. This is actually a very important contribution from Joan Massaguet, who has described the fact that TGF-beta turned on a very powerful growth arrest program, and he sorted out the mechanism of this. And basically, the story is simple. There is a FOXO transcription factor that is an adapter and partner of the receptors meds and SMAD4 for P15 and P21. These are CDK inhibitors, and therefore, you inhibit cycling dependent activities of these kinases, and therefore, cells accumulate in G1 and do not enter into S phase. This is how they make a growth arrest. Another famous example, and very fashionable these days, is the capacity of TGF-beta to turn on an epithelial to mesenchymal transition. Now, this is, of course, very interesting because it's connected to the control of cell polarity, stemness, metastasis, etc. And here, TGF-beta is a very powerful and profound inducer of EMT through regulation of snail and ZEB. Now, we don't know the partner for snail transcription. We know that for ZEB, it's ATS transcription. The ZEB and snail then are repressor of icadirin and repressor of MIR200. And these two genes are of epitheliality. Icadirin and MIR200 basically are the main determinant for a cell's being epithelial or not. And so this is how TGF-beta basically intercept the molecular pathway that are essential for keeping an epithelium an epithelium. In the third part of my talk, I would like to tell you about how the TGF-beta pathway gets corrupted in cancer. And this is very well explained by this slide. As you can see, on the left side, we have TGF-beta in normal epithelial and early stage in tumors, in which tumor suppressor activities dominates. Now, of course, this is a friend on this side, promoting, as I told you, growth inhibition, apoptosis, genomic instabilities. But now you see a different paradigm. The same cell, same history thus. Once it becomes tumoral, changes completely its responsiveness to TGF-beta. Now this has the same embryology, of course, and likely same context, but clearly the TGF-beta pathway changes completely, and it becomes a very profound pro-metastatic inducer, inducing EMT, invasion, survival, as well as profound effects on the tumor stroma, such as tumor suppression and angiogenesis. So how can we explain this one now? Clearly, there must be changes in genetic and epigenetic context that changes TGF-beta responsiveness from a friend to a foe. So we have been intrigued in searching for these, and basically finding, I think, of the lab was the idea and work by Michelangelo Cardinal that has linked TGF-beta signaling with the p53 family members. So basically what Michelangelo found by complete expression cloning. So it was not that we looked for p53. So people, when we started this, and this was thanks actually to Giannino, without his help, many of these initial experiments could not be carried out. They told me, you must be crazy. You know, we were coming from the embryology. I said, how do you want to work on such a stupid protein? Half of the world is working on it. How could you find something new and interesting? But Michelangelo correctly told me, well, no, we didn't look for p53. It was p53 that looked for us. So perhaps it's worth giving a better look. And in fact, I think he was right. So basically what he found is that p53 was an essential input in the TGF-beta cytostatic program. Without p53, TGF-beta growth arrest is not happening. And the reason is that basically p53 and SMADs form a complex, and the complex jointly regulated a set of target genes that are relevant in embryos and are relevant for growth arrest, including, for instance, p21. Now, this complex that, of course, is relevant for 53, but only for 53 family members, is not constitutive, but it is regulated. How? Well, you will know that p53 is activated by DNA damage, for instance, but I would like to underline that what we discover is that there is a perhaps more physiological pathway for 53 activation that leads to phosphorylation of 2 N-terminal serines, and this is mediated by the RAS-ERK CK1 pathway. And only when this is phosphorylated, the system lock in place with the SMADs and turn on TGF-beta growth arrest target genes. And I think this is a very nice way, you know, to control how FGF or oncogenic mutation of RAS can crosstalk with TGF-beta signaling. And our hypothesis is that, in fact, this is one of the main mechanisms by which, you know, it's the last resource for cancer cells to keep control of their proliferation. We will know that RAS is also an oncosuppressor to some extent, and it is an oncosuppressor only until p53 is there. And once p53 is gone, then RAS unleash all its pro-proliferation capacity, etc. And I think this is part of the loop that we described, in which RAS turn on p53 activation that raise sensitivity to TGF-beta and basically balance the pro-proliferation capacity. And so this has, I think, relevance with oncogene-induced senescence, etc. So suggesting the status of p53, wild type or null, or its phosphorylation status, may modulate tumor suppressive versus mitogenic effects of RAS. So we have been describing something happening here on the left side of the slide. Now let's look on the right side. Is there p53 relevance there? And actually, if I do this little trick to the slide, I would not make such a big mistake. Only that I need to exchange TGF-beta here with p53, which, as you well know, it's a very potent tumor suppressor on this side of the slide. But once I add the mutant p53, well, this is also true. Mutant p53 is a very powerful pro-metastatic factor. There are mouse models from Gigi Lozano and Tyler Jack showing this. So while, for instance, p53 loss can make tumors, mutant p53 knock-in mice make tumors that are metastatic, very aggressive tumors. Now, coming from the TGF-beta field, from this slide, you can realize how, for us, was quite remarkable the analogy between the two worlds. We had been describing this pathway with Michelangelo. So together with Maddalena and Orno, what we did was perhaps the mutation of p53 is changing TGF-beta, is part of the metastatic switch in TGF-beta. So just like they were cooperating in inhibiting growth, perhaps now they are cooperating for migration, invasion, and metastasis. Wild type RAS or oncogenic RAS were working with wild type. Well, perhaps oncogenic RAS, well known to be essential for metastasis, is now operating with mutant p53. Why not? Made perfect sense to us. And in fact, we made the experiment. So So we knock down mutant p53 in aggressive breast cancer cells, metastatic. And already in matrigel, what you can see is that these cells start retracting their filopodia. They look much more benign on a very microscopic surface view. They are perfectly fine. Actually, if you inject, they grow as tumors just okay. But in vitro, they migrate much less. And in vivo, they have lost their metastatizing capacity. This was quite shocking because this was such a simple experiment, and this had never been tested before. But this pointed, the mutant p53 is selected because it's empowered metastasis. Now you may ask, where is Tgf-Beta there? Tgf-Beta is everywhere. And certainly I could go with this type of slides for the rest of my talk. Just there's a huge amount of literature telling you that Tgf-Beta is autochtonally produced by advanced breast cancers and many other cancers that are also particularly responsive to environmental Tgf-Beta. So whenever you have a breast cancer in advanced stage, this is its motility and metastatic capacity is driven by autocrines meds. This is a huge amount of data showing and supporting this type of things. So how might this work? Well, this is the big mystery in cancer field. Mutant p53 is inactive transcription factor. It's a dead transcription factor because mutation invariably hits the DNA binding domain of p53. Why cells would like to keep a dead transcription factors? Why do they select for it? So we would say that it is because it empowers a program and that these programs include metastasis capacity. How does it work? Well, this work was initially suggested by work from Giovanni Blandino, who was the first actually to propose that the core domain of mutant p53 can actually have affinity to p63. Now we fully support, in fact, this as the most fundamental event by which mutant p53 operates in cancer cells, which is by inhibiting p63. Now, of course, this is a chemical titration. As you well know, if you remember your chemistry classes, titration is a very bad way to inhibit because you go from logarithms. It just never ends. There is no way for the mutant p53, no matter how stable it is, to oppose in an epithelial cells, it is loaded of p63, to oppose it by a simple titration. So what you need is actually some help. And this is what SMATs do. They basically make this complex quantitative. They titrate out p63 in a very strong complex, and this is where RAS again starts to kick in. We think that the TA domain of mutant p53 and the alpha domain of p63 are relevant. So basically you make a complex, and this is how it works. So p63, in our view, is the most downstream element of the Tgf-Beta mutant p53 pathway. So I would like to tell you something about migration, but I could show you similar data on metastasis. So basically, this just follow on the black bars here. If you knock down mutant p53 in these aggressive breast cancer cells, cells do not metastasize and do not migrate anymore. But now, if you concomitantly knock down also p63, then metastasis and migration comes back. In other words, you really do not need mutant p53 to empower migration as long as you don't have p63. Epistatic, we say that p63 is epistatic to the pathway. Yes, done. So p63 is a metastasis suppressor. Overexpression of p63 inhibits completely metastasis, and this story was then validated by work from Karin Vausden and Elsa Flores, who showed that, in fact, knockout of p63 is inducing very aggressive and metastatic carcinomas. So we think there are several roads leading to p63 inhibition. We described p63 metastasis suppressor through the mutant p53 pathway However, I believe there will be many other mechanisms, microRNA, post-translational, whatever, transcriptional, actually, very important transcriptional stories that inhibit p63. So you may ask, why do you need to inhibit p63, and why do you want to mutate p53 and not take away p63 directly then? Well, the reason is that p63 is also important. This work from Frank McKeon and Alia Mills from sustaining stem cells. Now, of course, losing p63 would empower metastasis, but these cells are not going to go very far because they are not stem cells anymore. Now, mutant p53, you catch two pigeons with the same stones because on one end you block metastasis, protection, empower metastasis, but also by some other mechanisms, you, I would say, sustain proliferation capacity. Finally, let me tell you where we are going with this. This is work from Marco and Elena. We are trying to understand how p63 is inhibiting metastasis. And this is basically the concept. We believe that metastasis can be a default mechanism, a default event. A default that is normally prevented by p63. Now, if you take the Tgf-Beta gene expression program in concert with environmental oncogenic cues, you know, there is a very complex program. Hundreds of genes related to metastasis can be regulated. But normally, the exploitation of this default program, if you want, is prevented by p63 and two target genes downstream of p63 that are sharp in cyclin G2. So while this is very complex, the metastasis suppression is actually very simple. And why? Well, I think that this is relevant in breast cancer patients. This is Kaplan-Meier, showing that tumor cells that have very high level of p63 are protected against relapse and metastasis, whereas those with low p63 are displaying a much worse prognosis. And so this is my last slide, telling you basically there are two arms. One arm is turned on by Tgf-Beta and a variety of inputs, inflammation, oxygen, hypoxia, stiffness of the matrix, cytokines, as well as cell-autonomous, oncogenic RAS, MET, RTK, whatever, your favorite genes here, that feeds on keep the flame of a pro-invasion metastasis program, but normally this is prevented by p63. The reason why Tgf-Beta is such a high profile, to end my talk, cytokine in this world, is that because it is not only taking care of this branch, but also in preventing the left branch. So it has an effect on both branches. So not only fostering this, but blocking the natural breaks. And on this scenario, I'll be happy to take your talks, to your questions. Thank you so much.